Here is an updated take on Picorna virus replication. So, in the Picorna virus genome, there is a small element that can fold up into a stem loop, and it's called the cis-acting replication element. CRE, cis-acting, CIS, acting replication element. In different viruses, this element actually moves around. So there's one virus where it's in the 2C region. There's others where it's in 2A. There are others where it's kind of all over the place. I think one it's up in the capsid region. So it doesn't have to be any place in particular, just has to be there somewhere. This is an element that can be bound by the 3CD polymerase. And protease. So remember, 3C is a protease, 3D is a polymerase. Here's a little structure right there. So at the bottom, these two little beta barrels, that's going to be the protease. And this great big thing, there's our little beta strands at the bottom. That's the palm domain. And we got our fingers over here, and we got our thumb as these 4 alpha helices right there. So it's like a baseball mitt that's like looking like that. Now it's coming around in a way that my hand won't turn. And there, that's the palm of it. So that's the polymerase, that's 3D, and this is 3C, and there's a little spot right between 3C and 3D that can be cut by a 3C. Thing is, the cleavage site of 3C is over here, and so it's hard for it to reach around and cleave behind itself. It's like trying to scratch that spot in the middle of your back. It's hard for us old people. Yeah. And why not pause for a moment for today's Easter egg? Because I've been forgetting to put these in. If you would like, for extra points, find me the largest RNA virus genome and tell me how big it is. Now, depending on when your information is from, the answer may be different. But there is one that is currently the largest, at least as far as I know. And you know what? If you find one larger, you'll get the points for that, for sure. Yeah, so have a go. Have a look. All right, back to the story. The cis-acting replication element can fold up, and it's assisted in this folding by 3CD. So 3C and 3D that can't quite scratch that itch, can't quite separate themselves. Look at that little string. You could just pull them apart. Oh, yeah. And this is going to be the site where we start primer synthesis. All right. So the primer is going to be a protein, and the protein is 3B, but it also has the name VPG, which is virus protein associated with the genome. Yeah. The V for virus, P for protein, and the G is genome associated. The G does a lot of work, even though it's lowercase. Yeah. VPG has a tyrosine, shown right here, at amino acid number 3 in the poliovirus version. And that tyrosine is absolutely conserved in all of the known Picorna viruses. So we think they all work the same way, even though it hasn't been demonstrated for all of them. Tyrosine has a nice little ring here, a little aromatic ring, and at the end of the ring there's going to be a hydroxyl, an OH group. And when we're adding nucleotides to other nucleotides, for example, remember we always add them at the 3' hydroxyl. That's where we join the 5' phosphate on. And so the 3CD polymerase. So 3D can't do this on its own. It's got to have its friend 3C along. The 3CD enzyme will add a phosphate, or rather, it'll add a U nucleotide, uridine, and it'll attach it via the phosphate to the hydroxyl group at the end of the tyrosine. Yeah. So it's a weird way to do it. It's definitely not normal, but that's how we're going to generate the primer. So let's take a look at a little bit more of this stuff. Here are some of these cis-acting replication elements. And we're just showing the top of it. Sometimes it's a longer stem loop structure. But the thing that they have in common is that they all seem to have two little A nucleotides right about that position. So somewhere between like 7 o'clock and maybe 10 o'clock or 11 o'clock, if you think of it as a little clock. Those A's are going to be important. So the mechanism is going to be that the 3CD polymerase is going to pick up a VPG. The VPG is going to align itself with the cis-acting replication element, and it's either 3CD or 3D. Now, I think the evidence is actually better that this should say 3CD is doing this, but yeah, the people that made this paper are very knowledgeable, and so we'll see. But we know you need 3CD and a little bit of 3D to actually replicate the process. Anyway, the polymerase will then add in a U nucleotide, so it'll line up here with a VPG, and it'll add a U nucleotide that's sort of held there by Watson-Crick base pairing with the A nucleotide in the loop. And so it's going to put in that U on A number 5, and it's actually going to slide it up so that U is now attached to A number 6. So it's going to pop it loose, move it up one nucleotide, reattach it, and then once again at A number 5, it's going to add a second U nucleotide. And so it's going to go from VPG to VPG with a U, and then VPG with a U and a U. So we're going to call that VPG PU and VPG PU PU, just like that. Okay. So here it is in words, then we'll do it in pictures after. And remember, this is the enzyme right up here that's actually doing the work. 3CD is needed, and a bit of 3D is needed. And the roles of these are not as well worked out as I would like. Yeah. So they're both in there together. And so right now I think we can definitely say that 3CD binds to the cloverleaf and helps to make primers. And we know that 3D definitely extends the primers. We're not sure if 3D also has a little bit of a role in helping to make the primers. So that's the little open question that's here, but it's not too big an open question as they come. All right, so in words, first, as we do every time, we're going to make a circle out of the genome of our virus. You will see this happen over and over and over again in pretty much every linear virus. They're going to start out by getting both ends together and making something like a panhandle. Now, in the case of Picorna viruses, the binding of those two ends together is going to be assisted by poly A binding protein, binding to the poly A tail, and poly C binding protein, which there's a tract of a lot of C's up near the 5' end of the genome. And so the poly C protein is here, the poly A binding protein is here. They're going to wrap around, and between themselves and the RNA, they're all going to interact and form something like a panhandle. So it's getting the same job done as something like a coronavirus that'll have, direct binding of the 5' and 3' termini. It's just doing it with some protein assistance, which is probably also true for coronavirus. We just don't know the names of all those proteins yet. All right, second, 3CD is going to bind to the cis-acting replication element, and it's going to pull it up into that little bunch, that little hairpin, just like that. And it'll grab onto it so that hairpin stays there, so that the two A nucleotides are in the right position for adding to the primer. Then either 3CD or 3D, here we go, or 3D is going to add two nucleotides to the VPG. And so that happens one at a time, and in between it's going to slide back. So let's say it'll add one nucleotide to the VPG, and then it will slide back and add by one nucleotide and add another U, and there we go. So that's now much more correct. Yeah, and that's just what we showed. 3CD will at some point be processed, and this is probably going to happen when another 3CD, like they probably can scratch each other's back. They can probably cleave each other, but they probably have great difficulty cleaving themselves. And so eventually you make enough copies of this that they find each other and activate each other. And from this point, 3D polymerase we know can use primers, but 3D polymerase can't make primers by itself. It needs to have 3CD there to make primers. Yeah, that's how it's currently understood. So that was step 5. 3CD is processed, meaning cutting the protein 3CD into 3C, protease, and 3D, polymerase. And remember, your 3C is down here at the bottom, your 3D is up here at the top, and that little stringy bit right there, that's where it gets cut. All right, and this primer, the VPG, which is also 3B, so originally it was just right before this 3C, it would have been right there. That is going to be the universal primer, and that is going to prime both positive strand and negative strand synthesis, which is cool. Yeah. And so this works because there are 2 A nucleotides at the 3' end of the minus strand, and there's a poly A tail at the 3' end of the plus strand. So either strand, there's a couple of A's. And a couple of A's is all you need, because it's only going to be 2 U's on the primer. And so, yeah, you copy your 2 U's and you're good. So to make plus strands, the structure at the beginning, which is actually the same structure that contains the IRES, it's also called just the clover leaf structure. They always draw it as like a little, 3-sided sort of clover of stem loops and hairpins, those sorts of things. It's going to have 2 functions. The one is going to be IRES function, direct ribosome binding. That's great. Whenever there's a ribosome around, it'll just clamp onto it. And the other function is going to be specifically interacting with the viral RNA polymerase. So the viral RNA polymerase is going to bind to the strands. So remember, after we start out with a plus strand and we copy a negative strand. So now we got 2 strands, which is great. But in order to copy these again, we need to peel them apart, and then we can make more plus from the minus and more minus from the plus. Yeah, I mean, that's what viruses do. They go viral. They make lots of copies. So to separate those 2 copies, or at least to separate the ends of those 2 copies, the viral proteins are going to come together and they're going to bind to the clover leaf. So they'll peel the 5' end of the plus strand off, and they'll also bind to the clover leaf. It's kind of the opposite of a clover leaf on the end of the minus strand, and so they'll peel them apart just like that. So the virus proteins that are involved here are 3CD, 2C, which is the helicase-like protein, and 3AB, 3B being the VPG. Yeah, I see that passed a couple times. Yeah, and you'll sound smart. 3B's the VPG, no problem. Yeah, there are also the same host cell proteins. The poly A binding protein is going to bind at the poly A tail, and that'll peel that off from the poly U that was made from the primer. And the poly C binding protein is going to help to bind the clover leaf and pull that apart. So now that we've separated the strands, now, we're going to put that VPG with 2 U's on there. The P here, like VPG, PU, just means it's a phosphate linkage, like a normal linkage. Since we're dealing with proteins stuck to nucleotides, it's, we need special language. Contrast that with coronavirus NSP9. So that case, we're actually adding on to not a hydroxyl, but we're adding it on to the amino group at the actual amino terminus of NSP9. There we go. Yeah. Yeah. And we're going to put a backwards G on there so we can cap it. And so it's going to be a little bit different. That's NERAN, that's coronaviruses. We're here to talk Picorna viruses right now. So VPG, little protein, protein 3B, with 2 uridines is going to be transferred to the 2A nucleotides at the 3' end of the minus strand. Those will then be copied, and that's where we'll line up. And so there will be proteins, probably 3D protein, involved in positioning this, because there are lots of places where there are 2A nucleotides in a row in this genome. It's like pretty common. So there are extra sequences around there. There's a whole cloverleaf structure. These are going to help to get the right proteins and the right primer into the right position. It's all kind of a machine. It's designed to trap all the right components so that they can then make copies of the virus genome. And then the interesting part, and probably the part that most people will remember, is 3D then just extends the primer and copies the whole thing. So just like in minus strand synthesis, 3D is going to do all the work. It's going to make the copies of things, but 3CD is going to be needed at least to bunch up the clover leaves and to make the primers. We definitely need 3CD for that, in addition to a little bit of 3D. So there you go. All right, hope that helped.